All right, we're picking up here on the next video. So we talked about drug design and just affecting receptors or affecting enzymes. And then if you're going to design something to kill a bacterium, bacteria have different life cycles. So things like that would disrupt a cell wall would be a good choice for bacterium. Of course, some bacteria have some more biochemistry, so sometimes you can get side effects because molecules will bind to the same thing. Okay, viruses are very interesting. So viruses, to correct this, they are nucleic acid. It could be DNA or RNA surrounded by a protective shell. They actually, it's kind of interesting to say whether they're alive or not because they can't actually copy their own nucleic acid. So they're actually a parasite. Okay, and they have two different phases. And I'm going to talk about viruses also. We'll get into COVID a little bit, and then we can talk about that when we look at drug design. Right, so this is a typical viral structure. You've seen pictures of the COVID virus. Looks a little bit different, but it's basically a protein coat and nucleic acid. And again, it can be DNA or RNA. For example, COVID-19 is an RNA virus. Okay. Now, what viruses do, there's actually two life cycles. Let me just see what the next slide is. All right. So what viruses do, a bacteriophage is a virus that affects bacteria, is they bind to the cell, the host cell. So if this is a virus that affects humans, it would bind to a human cell. So something like COVID-19 binds to respiratory cells in humans. All right, cells in your respiratory system. Once it binds, it injects its nucleic acid. All right, so now this is the DNA's nucleic acid, and of course this cell can copy nucleic acid. Now, in an active infection, it's called a lytic cycle. What happens is the host cell makes copies of the nucleic acid for the virus and actually makes viruses which assemble, and then the cell fills up with viruses to the point where it pops and a bunch of viruses come out. Right? It's called lytic, so the term lysis, to break apart, right? You've heard of a product called Lysol, right? It breaks apart things. Okay, so lytic means it's at literally breaking cells. And of course, the reason you're sick when you have a viral infection is first you have an immune response against this, which we'll talk about. But these cells are dying, they're being killed, so there's cell death. And of course, these cells have a function. And there's a lot of debris and a lot of liquid that forms as well. Okay, this is an active infection. Some viruses can do what's called a lysogenic. So again, remembering we talked about roots of words, lyso, right, breaking apart, genic, giving rise to. So this is an infection that where you're actually not sick. And what happens is the viral DNA or the viral nucleic acid incorporates itself into the host nucleic acid and it just sits there. And then something can trigger it to go into a lytic cycle. What can trigger it? A different infection, stress, things like that. Some examples of lysogenic viruses, herpes viruses. So cold sore is a herpes virus that does that. And of course, it's called a cold sore because you get a cold or a secondary infection, and then you start getting this kind of thing going on. And these can sit there for a very long time. The AIDS virus does the same thing. So it's tricky because you can't really kill a virus because they're not really alive. Right? You can't like disrupt its lifestyle. So what you have to do to design a drug is they have to interfere with some aspect of the viral life cycle, with the attachment to the cell, with the assembly, with the copying of the nucleic acid, something like that. Now, just a little bit more. When a virus gets in there though, it doesn't just stick to any cell. It binds to a protein on the surface of that cell, and it has the same idea like a ligand and a receptor. And all cells have proteins on their surfaces, and those things have a role. It can be as simple as cells sticking to each other, cellular adhesion. So viruses that cause the cold will stick to some of those. They can be involved in signaling. And viruses bind to those proteins as a first step. And when we look at the end of this, you'll see specifically for COVID what it binds to. I think I have that in a slide. All right. Viruses also have proteins on their surfaces because they basically are proteins, but they can have little spikes sticking out. This is COVID. So when you get a viral infection, your body fights it off. You can't take an antibiotic because they're designed for bacteria. So I'm going to explain how the immune system works. All right, so this is a diagram. So these numbers here are going to correspond to some things I talk about in the next slide. I'm going to go back and forth. All right, so the antigen is the term for the invader, the thing that's coming in, whether it's It could be a bacterium, it could be a virus, it could be a toxin that comes into your body, like say from botulism or tetanus, right? And what happens is it's picked up by a white blood cell called a macrophage or a B cell. That's shown right here. So here's my antigen, and this could even be a vaccine, and it gets picked up by a cell, and these are white blood cells floating around in your body, all right? And it gets picked up by that, and that's what those cells do, is they just pick things up and they eat them and they chew them up into little bits, and then they stick them out on their surface. Step 2 is that this antigen is presented to what's called a T helper cell. In the old days, we called them T helper cells. Now they're called CD4 cells. All right, and it's presented to it, and it basically says, here it is, here it is. So going back, that's this step. So the antigen, the little pieces of the virus or the bacterium or the toxin are put on this molecule here on the surface, and it makes a shape, and then it has a recognition event, a binding event to what's called a T helper cell. Okay. This is the cell that the AIDS virus destroys. It's kind of like the Paul Revere, if you will, of the immune system, because what the T helper cell does is it sends signals, hormones basically, to other parts of the system. It stimulates what are called B cells to reproduce and make antibodies. It stimulates cytotoxic, so cyto means cell, toxic, to kill, right? It stimulates these cells to reproduce and basically blows up the immune system and says, okay, it basically sounds the alarm. And these things will cause B cells to make antibodies. It'll cause these things to make things that kill other cells or will kill the antigen. All right. Antibodies, what they do is they bind to the antigen and then they make it more tasty to get picked up. It's kind of like sugarcoating it. Okay. So that's what happens in an immune response. And when you're sick, a lot of the symptoms you have are actually your body's immune system. Also, why are you tired? There's a huge amount of cell division that goes on here. Cell division takes a lot of energy. Making proteins takes a lot of energy. That's why when you're sick, you're tired, because you're actually using a great deal of energy. Also, a lot of the goo and stuff that you feel like when you have a cold, a lot of cellular debris, a lot of liquid gets pulled in there for this to take place. All right. So let's talk about vaccines for a moment. How does a vaccine work? Right? What a vaccine does is it is typically a piece of, let's just use a virus as an example. It might be a piece of the virus. So if you back up to here, right? Let's say this is a virus. And what happens is the virus comes in and it gets picked up by the antigen presenting cell, which chews up and takes little pieces of the viral protein or pieces of the virus and sticks them here. So what you can do is in a lab, you can take the virus and you can pull its guts out, so to speak, and just take the proteins from the virus and inject them, or kill the virus and inject them. And then it causes the immune response where you get T cells activated, B cells activated, antibodies, and so on. All right, and now you're prepared to, you can blow this up much faster. The problem here is that you have to have these B cells laying around, and typically you don't have very many. But I have a slide here that talks about immune memory. All right, so this is immune memory. So again, this is the antigen presenting cell. This is the antigen presentation and the T cell, T helper cell. And then what it does is it makes B cells, they're also called plasma cells, that will fight off the infection. But if you're not really getting the infection, so normally let's say normally you have, I'm just making up a number. Let's say normally you have 10 of these floating around. During an infection, you might blow it up to 1,000 or 10,000. After the infection is over, instead of keeping 10 around, you might keep 100 around. So that means to go from 10 to 10,000, instead you're going from 100 to 10,000. So that's basically how it works, is it's showing your immune system what could come at it, and then you have more, you're basically leaving a standing army behind. So that's how vaccines work. Now, the thing about them is that you are giving, people get freaked out. They say, well, you're giving me the virus. Well, typically it might be a virus, but it's inactivated. So it might have been heat killed, or it might be a piece of the virus. And so you actually can't get sick from the vaccine typically. It could be weakened. Okay. But there are some safety issues, all right? There's always a risk for an allergic response. Some people can, that's why when you get a vaccine, especially when you have your children vaccinated, you get a vaccine, they will tell you Keep an eye on things, watch for like a rash or shortness of breath, things that are an allergic response, and come back to the doctor very quickly. So some people just have an allergic response to the vaccine. There had been a big concern about mercury, and actually it's not really a big deal. It was contained in what was called a preservative. The amount of mercury in a vaccine was about the same in a 3-ounce can of tuna. The toxicity is due to methylmercury. Thimerosal is actually made to ethyl mercury, which doesn't live nearly as long in the body. And you can read through these two websites, all right? What's happened because of the uproar about vaccines, which was based on stuff that just flat out wasn't true, vaccines do not cause autism. That was proven to be false. That was a false paper, actually. But what the medical community has done, or the companies that make vaccines, they used to make vaccines in multiple doses so you could give it to a couple of different people. Now they make it in single dose, so they don't need a preservative. They just throw it away. So they don't contain the mercury anymore anyway. Right? Now this is a really detailed article. I'm going to spend just a few minutes on this on COVID-19 because we're almost done with this, and then we'll segue into this. So this is an article. So this is what this says. It's an RNA virus, targets cells through the viral structural spike protein and binds to this particular enzyme on the cell surface. So remember we talked about enzymes, proteins on the cell surface in case it binds to this enzyme, which is on cell surface. So they've already found this out. And this is how the COVID virus works. It binds to this protein, then here's the nucleic acid coming in this case RNA. There's a process where it's made into viral proteins and reassembled and put back out. Each of these steps are steps where people are working on drugs to try, or vaccines to try to make this work. So for example, you could take just this protein here and make a vaccine from that, and that couldn't make you sick. You could take the vaccine and heat it so that the RNA is inactivated, then you couldn't get sick from it. Those are examples of how we do it. Okay, so that's where we'll pick up for drug design, and that's the end of this set of slides. There we go.